let's have a look at metabarcoding data as an overview, sort of. So, why metabarcoding? So, why should we go and do this type of sequencing? Well, the idea is that we use this to find the composition of microbes in some microbial community. Composition, and that is actually important phrase here. And the point is that with metabarcoding we can find this composition even if the organisms are unknown. And this is the big sort of benefit, I would say, of this method. You can study the new environments in a way that you cannot do with full metagenome sequencing. So what is sort of the pros and cons if you compare this metabarcoding versus the full metagenome sequencing? Well, here are some. Plus is then exactly this. We can map into unknown terrain. So when you're studying environments or microbial community that hasn't been studied very well before, this is the natural first step to take, metabarcoding. It's cheaper per sample, which means we can sequence more samples. And this is of course also important when you dig into new terrain, right? It's much more important to get an overview than to study in detail some specific part of it. That can come later. The data processing is also simpler compared to the full metagenome sequencing data. This follows mainly because there are fewer reads or smaller samples, right? So I'm not sure if it's actually the steps themselves are simpler, but at least this is how people tend to think. A disadvantage of metabarcoding compared to the full metagenome sequencing is lower resolution. You cannot study the sort of the very fine resolution composition, or you cannot study single strains, right, with this technique. And of course you don't get any information about the genomes actually, because what we will do now is to sequence a particular marker gene, one single marker gene, not the whole genome. That's the whole idea. So if you want to understand more about the genomes, the genes they have, the proteins they encode, and so on, then you have to do full metagenome sequencing. There is no way around that. Metabarcoding is for getting the overview of what type of organisms are there and how abundant are they. So if you look, if you remember this sort of picture here from actually week 1 lecture, these are all examples of sort of metagenomics, right? You collect DNA from all kinds of strange or not so strange environments, and you do sequencing of this and some bioinformatics. And this applies to both, right? It's all about metagenomes. But what you actually sequence is different. So with full metagenome sequencing, you dig deep into the samples that you take because you sequence everything, right? But with metabarcoding, you instead covers wide. So you only sequence one single gene typically from these samples, but on the other hand, you sequence many samples. So the focus are quite different here, and what you can achieve is totally different. You could argue and say that, okay, everything that you can obtain by metabarcoding, you can also obtain by full metagenome sequencing. And in some sense, that's true. So if you have sort of infinite amount of money and resources, you can do full metagenome sequencing of lots of samples. But that will, as for now at least, this will give you an enormous amount of data and horrible analysis time. So this is sort of a quicker way of achieving the overview, you could say. So when you sample the microbiota, and notice I use the phrase microbiota here now, not the microbiome. And I think there is no sort of real rule here, but I think there is a consensus that we use the phrase microbiome when we do full metagenome sequencing, because a biome is the collection of genomes, right? We use the phrase microbiota when we only sequence marker genes, because then it's no longer genomes. So we shouldn't call it microbiome, but instead microbiota. So the microbiota contains an unknown number of an unknown organisms in an unknown mixture. So this is sort of the headache, right? We don't know how many organisms are there, we don't know which organisms are there, and we don't know how much there are of each of them. And this is always the case, right, in all metagenomes. And if you remember when we talked about metagenome assembly in module 8, we saw that if you have a community, you can only sort of hope to reconstruct the genomes of the most abundant organisms, right? Those who come with low abundance, low coverage, they will not be assembled. We cannot reconstruct those. Here we can see all of them, also the low abundant ones. That's the point of doing metabarcoding, you could say. So we do a targeted sequencing of a specific marker or markers, which is also why we sometimes call this amplicon sequencing, because before we do sequencing, we amplify with PCR a certain region or a certain gene, and then we do sequencing of these amplicons, the amplified part of the genome. So we don't sequence the whole genome anymore, only these very specific markers. So assume we have now some genomes here, and I draw them as circles because bacteria genomes are, the DNA is usually organized as one single chromosome, which is circular. So let's say we have 4 types of bacteria here now, and the 4 colors indicate that. So let's call them just green, yellow, red, and blue. In shotgun metagenome sequencing that we have seen up to now, we would now cut this into small pieces, all of them, and sequence the pieces, right? So we get the reads. And then we try to puzzle together the pieces again in order to reconstruct the genomes. Yeah, this is not what we are going to do now, not at all. The point is, each of these genomes have a gene that they share. All of them have the same gene. Instead of sequencing the whole genomes, we now sequence only this gene from each genome. And here are the reads we get, and I have now sort of sorted them so that they are by color. And of course, in the mixture that we sequence, we don't know how many there are of each genome. There can be many green or few yellow or many red. This is sort of like you have a sort of a bowl, right, with small balls in it of different colors, and you don't know how many you have of each color, but you sample. You sample, in this case 20 reads, and then you look how often did I sample the various colors, and from that you can now, of course, conclude that the relative number of reads from each organism reflects their actual abundance in the bowl, right? So in the soup of genomes, this is sort of the sample you get. So a composition of a sample or a microbial community is the same as the organisms that we find there and the relative abundances. So in this particular case, the composition would look like this. You see that organism red is 25% of the times of the reads, right? So we assume that our community contains around 25% of red, and 40% blue, only 10% yellow, and so on. So these relative abundances here, 2 out of 20, right? 0.1. That's the reason for these numbers. And notice they sum to 1, right? It's a relative abundance, because the total number of reads here is not interesting at all, because that's how deep we have been sequencing. That has nothing to do with how abundant they were in the first place, these organisms. Because remember, we did some amplification here. So we don't really see that original abundance. It's only relative. How much do you have of one compared to the others? Whether there were originally many or few bacteria, that's a completely different type of question. It's impossible to answer in this way. Okay. The 16S rRNA gene. This is the marker we almost always go for. And the reason for this is found in all prokaryotes. All genomes have this gene. It's also quite conserved, so it's possible to recognize it regardless of which organism, which genome you look into. You will always, if you have the complete genome, you will find the 16S gene there. It's not that different from one organism to another. They do differ though, and they differ enough to make it possible to separate between at least the genera. Species is possible to some degree, but too many species will have very similar 16S to make it a reliable resolution. But on the genus level, genus rank, we can pretty well separate. There is also very little horizontal transfer for this gene, so it reflects sort of the long-term evolution. There is some horizontal transfer though, and it's been reported lately that it is perhaps a little bit more than we originally thought. And that of course makes it a little bit noisy, but still it's considered a smallish problem. It's around 1,500 bases long, so it only covers a very small part of the genomes, right? But it's also known that it varies in how many copies you find in each genome, and this is important, right? At least to some degree, this is important. Remember from the previous slide with these 4 genomes, right, with the different colors. In my drawing there, I was drawing one single copy from each genome. The red, the green, the blue, and the yellow, they had only one sort of gene each, marker gene. This is not the case really for the 16S. It varies, right? So some organisms have 1 copy, some have 2, some have 5, some have 7. And of course, the number of reads you get depends also on this, right? So if one organism has 10 copies, another one has 5, and these organisms are equally abundant in your sample, then you will typically collect twice as many reads from the one with 10 copies compared to the one with 5 copies, simply because it had twice as many copies per genome. So this will then affect the composition, right, the number of copies. This is not really a big problem because the number of copies will be the same each time. So when you compare compositions again over, for instance, different treatments or conditions and so on, these copy numbers will always be the same. So if you only look at the change in composition, you don't need to take this into consideration. But if you want to sort of say something about how abundant the bacteria really are, then you must remember this. Yeah, this 16S gene also has what we call variable regions, and here is a figure from an old publication of ours. And along the x-axis here now you have the position, And notice this is more than 1500 positions. This is like over 3000 positions. So what is this? It's because when you do a multiple alignment of many sequences, and they all are around 1500 bases, you will have to add some gaps in the sequences, right? In the alignment. And that's why the length of the alignment is always longer than the single sequence, because of the gaps. So these are actually full-length 16S sequence aligned, and that's and due to the gaps, it's more than 1500, right? But here, and on the y-axis here, you can see entropy, which is sort of the variability in the bases on each position. So if you have a tall gray bar, it means the sequences are very different from each other at that position, or they vary in composition, right? A, C, G, or T. So where you have the largest bars here, you have the greatest variability. And you can see there are actually sort of 9 mountains of variability along the 16S gene. And these are the variable regions. We usually denote them V1, V2, V3, and so on, up to V9, variable regions. The point of this is that when you want to sequence the 16S, you don't sequence the whole gene. You sequence only part of it. And this colored bar here indicates where you have the sort of informative sites, as we call it. Where should you sequence in order to separate between different taxa? And you can see there are many informative sites, but note here this V3, V4 region is very two distinct mountains here. And here we have several of these very informative sites. So what we typically do, we amplify and sequence a piece of the 16S. So we don't even sequence the whole 16S, only a small piece of it. And how small depends on the sequencing technology, really, because with the long read nanopore, for instance, you can actually sequence the whole. But with Illumina, since the reads are shorter, we want a shorter piece of the 16S so that we can actually see the whole sequence. And typically the V3 to V4 region, and this is this part here. This part here. This is most common to amplify, but there are also other, some copy down here and some up here. So this varies, but this is the most common choice, you could say. The point is, with Illumina sequencing, short reads, you cannot sequence the whole because then your reads doesn't cover the whole. It must be a piece that is short enough to cover by your reads. So, the amplicon sequencing principle. First, you do PCR amplification of the targeted region. And for that, you need what we call a PCR primer pair. Let me try to illustrate this. So here we have the double-stranded genome, or a piece of it, right? And here is the 16S gene that we want to sequence now. Then we design primers, a forward and a reverse primer, which is sort of short pieces of DNA that actually matches exactly these two regions of the 16S. It matches exactly here, and it matches exactly here. And then in the PCR method, this region will now be amplified. This double-stranded region of the genome will now be copied, and it will be amplified. So it will, if you run the PCR in multiple cycles, right, it doubles for each cycle. So if there was 1 copy after 10 cycles, it's 1,000 copies of this, and so on. So we only see, or we actually copy this piece of the genome. The rest is lost now, right? So we are left with only this here piece of the genome. And primer bias. What do we mean by primer bias? The point is, no part of the 16S is conserved across all organisms. So these primers, right, they must match exactly, and they do so in many versions of the 16S, found in many organisms, but not all. So you may have some organisms in your soup, in your microbial community, that you don't really detect because the primers don't match their 16S good enough to do this amplification, and that is primer bias. And of course, lots of effort have been made to try to make primers that binds as universally as possible to as many organisms as possible, but you cannot really expect to bind to all organisms. So no really primer pairs are actually universal, even if they are sort of called that. If you buy from a company, they will say, oh, we have universal primers. No, they don't have that. I mean, no one have that because it's not theoretically possible, but you can have almost universal, right? You can have something that covers vast majority of organisms, but not all. And some organisms will then not amplify, or at least amplify poorly. So you don't get as many copies as you should from those, and then they will be lost more or less by very low abundance. So let's have a look at a couple of primers here, and this is there are many primer pairs to choose from in this world for 16S amplification. We will have a look at some of this in the module 10 document. What you should notice here, so these are sort of this is the sequence here that should bind here, right? So you have to know how the 16S sequences look like in this region to design this primer. Note that the letters here, R, B, and S, these are not bases. What are these? These are what we call the ambiguity symbols. And these you see frequently. So R actually means either A or G. So it means that the primer sequence looks like this: C, C, T, A, C, G, 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 R. And then you actually replace R by either A or C. So here you have a choice of two bases. And here you have B means C, G, or T. So here you have a choice of three bases. And S means also two, C or G. So it actually means that this single sequence that is sort of spelled out here represents in total 12 different sequences. Because you can here replace R with either A or G. And for each of these two, you can replace this one with C, G, or T. So you have 2 times 3 combinations, right? And for each of those 6 combinations, you can have also 2 here, C or G. So it means 2 times 3 times 2 is 12 combinations. So this actually reflects the primer. The primer is actually 12 different sequences. And these 12 are then used as primers here, matching all that these 12 sequences could match. And the same for the reverse. So when you see these ambiguity symbols, you now know what it means. It means that there are some sort of variability at that position, and the primer that you see as one sequence is actually representing more than one sequence. So the amplicon sequencing principle. First of all, multiplexing, that's a phrase that you meet. What is this? Well, this is purely convenience. Instead of, I mean, we don't need to sequence very deep when it comes to this meta barcoding. So on each sequencing run, in order to make the sequencing efficient in terms of using the resources efficiently, instead of sequence each sample very deep, we sequence many samples instead. And that's, again, the sort of the advantage of meta barcoding, right? Cover wide instead of deep. But how do you do that? Well, amplicons from different samples are then sort of marked with a barcode sequence. So this is sort of an artificial piece of DNA that we add to the amplicons. Technically, has nothing to do with the biology. It's just what we call a barcode sequence. It's a short DNA sequence that we add to the primers actually. So when the amplicons are sort of swimming around in our soup here after we have amplified them, they typically look like this, where we have those with the red barcode here are coming from the same sample. Those with the blue barcodes here are from another sample, and those with the green is from a third sample. And these can now be sequenced together because we have this barcode as part of the sequence, so we can actually identify from which sample was this actually taken and from which sample was this actually taken. So that's purely convenience to make things efficiently. So when it comes to the actual sequencing, we have now these amplicons from various samples. You can see by the various barcodes here. They are all amplified from the 16S gene, right? With this primer. The primer is the same in all samples, right? Primers are the same. So when we have one of these amplicons, we now do straightforward Illumina sequencing paired end, as we have seen before. An R1 read from this end and the R2 read from this end. And notice now this R1 read will sequence both the barcode and the primer and the rest of the 16S down here. But it won't take rest of the amplicon, I should say. This is not the whole 16S, this is only the amplified part, right? But then we sequence from the other strand, the other direction, also covering the barcode and the primer, and the rest of the amplicon down here. And then you can see these two overlap each other. So together, these two cover the whole amplicon. Together, they cover the whole amplicon. And this is important. So the fragment lengths are now very similar. Remember, we talked about fragment lengths when we did full shotgun sequencing as well. But then the fragments length varies a little bit, right? Some are short, some are long, because they are randomly chopped up DNA. This is not randomly chopped up DNA anymore. These are designed amplicons. We copy a specific part of the genome. And since these primers match more or less at the same part of the 16S gene each time, the fragments come out almost the same length as well. It doesn't vary very much. It's not 100% identical because, of course, the 16S sequences are a little bit different from one organism to another, but they are pretty similar in length. So we sequence with read length capital L, then we always design our primer such that the region that we amplify, the length of it, is less than 2 times the read length. So let's say we have read lengths of 300 bases. That's common for amplicon sequencing. Then this amplicon should be shorter than 600, right? 2 times L. And it should be actually quite much shorter too. So let's say it's 400. Then it means that we sequence the first 300 here and miss the last 100. But then we sequence those here, but we miss this. But the overlap will be 200 bases, right? So it's more than enough to sort of glue these two together into one long sequence covering the whole amplicon. That's the idea. So when it comes to the fragment length here, they're all pretty similar and they should not be long. When we talked about fragment lengths and for full metagenome sequencing, at least if you're going to do assembly, you should try to have a little bit long amplicons so that the reads don't overlap. Here it's opposite. We want them to overlap. So you should not have long fragments. Amplicons should be fairly short. But again, remember, if you do nanopore sequencing, long reads, you can have long amplicons. So overlapping reads is something we always want if we have paired-end reads. Read processing, yeah, that's what we are focusing on in this module 10. And that means filtering and trimming, etc. That's the first thing we do. And that is more or less the same as we did also for the shotgun reads. We can do some sort of filtering based on the quality scores and so on. And trimming of the 3' end. Why should we do that? Well, remember that the 3' end is always the most sort of error prone in Illumina reads. And when, as you saw in the previous slide, we want to merge the reads, or they overlap, right? So we want to sort of join them together into one single read. And then that is actually a little bit easier if you trim off the worst part of the reads. But you mustn't trim so much that you lose the overlap. The length of the overlap must be long enough, you could say. But so this is something we usually do it, but it shouldn't actually be necessary when you think about it. It has to do with how these reads are being merged in from two R1, R2 reads into one. And then the merging, from read pairs to a single read, covering the whole amplicon. That's with Illumina then, if you have paired-end reads. You can, of course, do meta barcoding with single reads as well. Then you have no merging step. And dereplication, what does that mean? Well, it means that since we now have many identical reads, it's silly to sort of store every single sequence multiple times. So you just store it once and then a number indicating how many times have I seen this read. So that's dereplication. It only means that you sort of count how many times you see each unique read. And store only the unique reads together with that copy number, you could say. Removing Chimera. So what is this? Well, Chimera is sort of an artifact of this PCR step. And it results in a read which is actually a mixture of two of the original sequences. And this is a sort of a display of this I copied from somewhere. So what happens sometimes when you do PCR of a sequence, so this is the sort of the sequence, and this is the copy that you want to make of it. Sometimes this copying stops halfway or something like that, right? For some technical reason or chemical reason, it stops here. So it doesn't end as a full copy. So this short one here will still float around. And then when the next PCR cycle is started, this one acts as a primer. Because it's a short sequence that floats around, right? Like a primer. And now, let's say this, so you can see this is a copy of this blue organism here. But this part of the blue organism also is the same as this part of the green organism. So this blue short sequence from up here is now used as a primer for starting to amplify the rest of this organism. So we use this and we extend it by copying this part of another organism, right? So you end up with a mixture, right? The read you end up with is partly from the blue and partly from the green organism. Hybrid, you could say. And this is what we call a chimera in this language, you could say. And of course, these sequences we should get rid of because they are not representing any of the original organisms. It's a mixture of them. So that's why we try to remove them. I have to say my experience is that this is not a big problem, right? The PCR chimera, not a big problem. Only a smallish number of reads will end up like this. But it's customary to try to remove them. So once we have done these steps, we have to do the clustering. And this is really the important step, you could say, in this processing. Because now we have all the reads, and we now want to the reads that come from the same organisms should be grouped into sort of a single group, right, or cluster. so that we can see how many reads are coming from this organism. And each group we now call an operational taxonomic unit, or OTU. So this is just a phrase that we use for these groups. Think of them just as they represent a group of reads that are similar enough to be merged together. Assume they come from the same organism. We will look at two distinct ways of doing it in this module. The first one is the classical one, abundance clustering. And what is the idea here? Well, first of all, compute the identity between the reads. How similar are they to each other? Reads with a minimum identity are clustered. So if they are similar enough, they are clustered into an OTU, right? Which means we need to have some threshold here, right? How identical must they be? 97% is a very common threshold to use, but this is something we can adjust. But they have to be close to 100, right? It's no point in trying to go very low here, because all 16S sequences are similar to each other. So if you go like to 75% similarity, then all will be in the same cluster, because all of them are that similar. So it must be close to 100. Most abundant read in an OTU is then used as the representative sequence for that group or OTU. And that is what we call the centroid sequence. It's just a phrase. It's sort of the center, right? It's not necessarily the center, but it's the most abundant, the one read that has the largest copy number. That's the one we choose as a representative, naturally, right? Naturally. The other approach is what we call denoising, which is also a way of clustering, you could say, even if the people behind denoise, they say it's an alternative to clustering, but that's just a battle of words, right? Because clustering just means grouping. In my eyes, it means it's not specific to some procedure. It means just grouping. And this denoising is also grouping the sequences, but it uses a different criterion. And it says that sequencing, the group only sequences whose difference is due to sequencing errors. Right? So the only reason for reads being different should be the sequencing errors. And this I would like to mention now. There are some good reasons for doing as we did up here, because even within the same genome, 16S sequences are not always identical. Remember, I told you that there are often many copies of the 16S even within the same genome, right? But these copies are not really copies. There are sometimes small variation between these, even if they are inside the same genome. So it means that looking for all variants of a sequence is a little bit too much resolution, I would say, because we want the reads from the same genome at least to be grouped together, and we typically want even bigger groups than that, right? Even the same species or even the same genus should be grouped together. And then there are clearly variation between them. But here we try to only look for the sequences whose difference is due to sequencing error. And in order to do that, we have to have some estimates of sequencing error rates. And that we can actually do from the same data. And the most abundant reads in the group are again used as cluster centroid. So that's the same between these two then. So actually, whether you run this first one here or the second one here, you end up with some centroid sequences, one for each OTU or group, right? So we have a FASTA file. That's the output from both this or this. But the FASTA files will be different. It's not the same sequences they find, but they will find sequences and list them in a FASTA file. Then once you have these, you can go and count how often do you see these various groups in each sample. And this is sort of the main data structure, I would say, the read count. And here you can see arranged in a read count table. So that's the sort of the second sort of output we get from both these two procedures. A table where you have the samples in the columns typically and the groups in the rows, and then you see count here how many reads from this group did I find in this sample, how many reads did I find from this group in this sample, and so on. And for each group you do this. So you have a table with rows and columns and read counts. So this is the number of reads. It's always integers, positive integers, or zero. Could be zeros, all right? This one was not found at all in sample 2, then it will be a 0 here. So the output is the centroid sequences as a FASTA file and the read count table as a straightforward table like this. Let's have a look at this abundance clustering idea. What is the idea? Well, order all unique reads by abundance. And then, I mean really all reads from all samples, right? Not sample by sample. Merge together or put all reads together in one big superset. Then order them, right? So that you have the most abundant read first, second most abundant read second, and so on. Use first read as centroid. So here we have, and these dots here now indicate reads, and the size of each dot indicate the abundance. How often have we seen the various reads? So this one we have seen most often since it's the biggest blob. And the distance between them reflects now the similarity, how similar they are, right? All other reads with distance less than d to the centroid belongs to the same OTU. And then we sort of remove these clustered reads from the list and start from the top again. So it means in this case, we would now start by selecting this as the centroid in the first group. And then we make this distance d radius circle around it and say that everything that is close enough to this one, that means inside the circle, is now member of the same group as this one represents. So we color them red, the red group, and then we sort of take these reads out of the list and we are left with only these two. Of course, in reality you have thousands of reads, right? So it's not that simple, but for this drawing. And then you choose this one as centroid because it's bigger than this one, right? This is the biggest now. And you make a circle again. And notice that this one is now inside the circle, but this has already been colored. It has already been grouped, so it's no longer part of this group. So this group here will only contain these two then, colored blue. And so on, and so on, and so on, until all reads have been grouped. So this is a greedy algorithm, as we say it, because you start out with the biggest one first. The biggest one can choose from the dinner table, right? Everything around it, eats all the other reads around it. That's one group. And then the second most abundant, which is not already, I mean the second was already here, so it was sort of taken by the first one. But then of the remaining ones, the largest one makes its choice, and so on, and so on, and so on. This is the classical way of abundance clustering. Let's have a look at the alternative, the Dada denoising algorithm, and that's slightly more complex. Again, we start with all sequences in the same cluster. Well, this is different, right? We start with all sequences in one super big cluster. And then we select the most abundant as the mother sequence. So that's similar to the previous step. We always start at the most abundant one, the biggest blob. We now estimate error probabilities from that given partitioning or clustering, grouping. And then we compute what is called an abundance p-value for all the unique reads other than the mother sequence. And if at least one of these p-values is too small, There's a threshold obviously here as well. Then we make a new partition or grouping, and then we choose, so we get sort of 2 mothers instead of 1. And then we choose the one with the smallest p-value as the new mother number 2, you could say. And then you continue on this. Yeah, and each sequence is assigned to its most likely mother. And then you continue until all p-values are large enough. So you create sort of new mothers, and each mother is a new OTU, right? Until you have so many mothers that all the other reads will be included in one of the groups with a p-value which is large enough. So what is this p-values, and what are, let's have a small look at the details here, but we are not really digging into the deep details. First of all, substitution probabilities. What is that? Well, we typically arrange them in a matrix like this, where the rows are where we come from and the columns are where we go to. Example, the mother sequence looks like this. This is a very short though, right? But for this example, here we have a potential child of this mother, meaning that this, we assume this is sort of the true sequence, while this has arisen as an effect of sequencing error. And we can see it has a difference here. C has become an A, and it's a difference here. G has become an A. Apart from that, they are identical, right? So there are 2 errors then, we can say, in this child compared to the mother. Now the probability of the child given the mother can now be computed, because we now go symbol by symbol through the reads. First, we go from an A to an A. What's the probability of that? Well, then we go from A to A, 0.95. So we write down 0.95, and then we go to the second position. We go from C to A. What's the probability of that? From C to A. That's only 0.01. Then we take the first value, multiply with the second value. Then we go to the third, going from G to G. Yeah, that's 0.95 again. And we multiply with this one, right? And then you proceed through the read like this. And then you can see that as long as you have identities, right, the values are big, typically close to 1, right? So you can multiply them together and you still have a pretty big value. But as soon as you start including these small values, the product drops sharply, right? So here we have 2 errors, which means we will have 2 differences, which means that we will have 2 of these small probabilities, this one and this one actually. So this probability becomes rather small, I would say, for this particular example. But if you have only one, yeah, it's not that small, right? You can see all probabilities become small, but some become super small. Now, what is what we call the abundance p-value in this setting? First of all, the mother partition has capital N members. So that means the copy number of the mother plus all its children. And remember, we start out with one single group with a sort of a super mother for all the other reads. So this is really a big number in the first round, but it becomes smaller and smaller as the number of groups increases, because then it's the size of each group, right? This is the size of each group. And then the child that or this potential child is now observed small n times. This is a much smaller value, right? So the p-value is now actually the sum of binomial probabilities using this probability that we computed up here as parameter. If you sum the binomial probabilities from this n, small n, up to the capital N. So that's how the procedure is in Dada. I think I would, when I look at this, I don't really understand Well, why do they sum here from small n to capital N? It's the same as 1 minus the sum of the same probabilities from 0 to n. So it's sort of the reciprocal of the probability of having up to n copies. I must say that I think I would have just plugged this into the binomial probability density and looked what is the probability at n copies, and use that as sort of the p-value. But okay. Anyway, you have to make a threshold on this p-value, so it doesn't really matter whether you compute it this way or the other. If a p-value is large, and that means you have to set a threshold here, then C can very well be a child of M. C can very well be a child of M. So, but if the p-value is small, it cannot be a child of M. And I can try to illustrate it here again. So again, we have these blobs that represent different reads, and the size of each blob is the copy number of that read. And now the distances here is not really how similar or different they are, but it reflects this probability here, right? So if you have a long distance, it's a small probability. It's a small probability, of having a child here compared to here. The abundance p-value become large if the distance is small enough and the size is small enough. So if we now start out with this as the mother, which of these other reads could now be children of this mother here? It could, for instance, look like this. I color them red. This is the mother, and all these others here are potentially children, but not this one. And why not this one? Because it's closer, right? It's more likely to see. It's not so different, right? So the probability here is quite the small distance means that her probability is quite high of going from this mother to this child. But the problem with this one is the size. It's too abundant, right? Because even if the probability of this error is quite high, it's not that high. It's not almost the same abundance as the mother. I mean, errors don't occur that often. So that's why this one is not now grouped together with this mother. Even if it's close, it has too high abundance. And this would never have happened with this abundance clustering, right? Because it only looks at the difference, the sort of the radius around the mother, and say everything inside the radius belongs to my group. All of you are my children, right? But this one is different because the abundance of this one, this cannot be a children of this mother because it's too big. So this will be a mother of its own, and then up here you will also have a mother of its own. So you get 3 different clusters, and this is sort of typical that you have many of these sort of loners in the denoising procedure. Okay, finally, taxonomic assignments. So from the entire data sets, we now have the centroid sequences, whether we do it by abundance clustering or denoising. We have a FASTA file, right? And we also have this table of read counts for each OTU in each sample. Now we can, of course, assign taxonomy to the centroid. Isn't that reasonable to do? I mean, we have their sequences. What kind of organisms are these? And that we typically do that. And this is sort of straightforward. It's a sequence lookup in some database with 16S having known taxonomy. So this is quite similar to what we actually did in module 9 with full metagenome sequences, where we have reads, right, from all around the genomes. We used this Kraken, or we used some Caillou or Metaflan. All these methods will try to take each read and assign it to some taxonomy. And it's exactly the same thing we do here, except that now all the reads are from 16S sequences, so we don't need to compare it to genome databases, we can only compare it to 16S databases. That makes it a lot easier, right? You can do this by k-mer methods or alignment-based methods. You can actually use the Kraken for this as well, as we did in module 9. And of course, as usual, the database you use is much more important than which method you choose, I have to say. This is typical for all taxonomic assignments, whether it's 16S or something else. But at least it's sort of straightforward, and we will not really emphasize this very much because some OTUs may be from new organisms and then we get no taxonomy, right? But they must still be important, and I think that we should not be too hung up on the taxonomy here, because when we do meta-barcoding, we expect to discover something new as well. Of course, there will be something which is already known to us, so many of them will be assigned taxonomy, but we must be prepared that there is also something new which is not assigned taxonomy, and it doesn't mean they are unimportant. On the contrary, these could be super important, the new ones. So we shouldn't be too hung up on the taxonomy. The read counts and the centroids are there. We should do the analysis based on that. And that's also what we will do. But this is all for now, and go into the module 10 document and read more about the details of the things I have been talking about and do the exercises.